Hello, everyone, and welcome to today's International Eosinophil Society monthly webinar. Today's topic will focus on eosinophil functions in metabolic health and disease. This is a very different topic than those that we've been covering since the beginning of the year, where we mostly talk about how eosinophils contribute to maintaining health, but also perpetrating disease in various disorders. We will hear today that eosinophils have other very interesting functions in adipose tissue and cardiovascular disease. Today's webinar, as all the other webinars that we've been organizing since the month of January, have been organized to continue fulfilling our mission, which is educating people about eosinophils and their functions, as well as eosinophilic disorders, but also bringing together scientists who share a passion for the functions of eosinophils. And you can, if you wish, visit our website, which is shown on the left-hand lower side of the screen. Just a few housekeeping recommendations. You will be able to ask questions, but not live. You can answer your questions in the question and answer button at the bottom of your screens anytime during the presentation, and our moderator will address these questions to the speakers. All of you will be muted throughout the session, throughout the webinar, and will not be able to use the chat feature. So really, you must concentrate on the question and answer button. If you have any technical difficulties, the best solution is usually to try to log off and log back on. And this webinar is being recorded. So if you've not been able to see all the presentations, or if you would like to suggest presentations to some of the people you work with, there will be a link on our website so that you can go and see this later on. I would now like to introduce today's moderator, Lisa Spencer, who is a longstanding member of our society and who put together today's webinar. She is associate professor of pediatrics, and she works at University of Colorado School of Medicine. Lisa, thank you very much. And now it's your turn to present the first speaker.
Good morning, everyone, or good afternoon, depending on where you are logging in from. So we at IES are really excited about this seminar. We're really looking at something that eosinophils are generally not traditionally considered to be a part of. So we're excited about expanding into some new areas. And I think as you'll see, the three speakers today span different countries, as well as some broadly relevant topics within this quite broad topic of metabolic diseases. So our first speaker is Dr. Alexander Eggel, and he is a research group leader at the University Hospital in Bern. And the topic he'll be presenting today is Eosinophils Restore Age-Related Adipose Tissue Dysfunction and Sustain Physical and Immune Fitness in Old Age. So Dr. Eggel, take it away.
Okay, well, thank you very much, Dr. Spencer, for this kind introduction and also for the invitation to be part of this very interesting webinar. It's a pleasure to be here, and as you see from the title, to talk about adipose tissue eosinophils and their function in maintaining physical and immune fitness during aging. Now, before we dive into our favorite topic of eosinophil biology, I would like to take a few steps back and give you a little background of why we think that it's important to study aging and how we came about to study more specifically eosinophil function in that context. Well, the simple answer to the question why studying aging is important is it affects every one of us, and so it is per se very important. Aging is basically defined as a multifactorial, progressive, non-reversible biological process that starts out with birth and ends with death. And during aging, our cells, organs, and tissue, they lose function due to the accumulation of mistakes and the loss of repair machineries. And on the other hand, our organism gets more vulnerable to develop age-related diseases and also is more prone to suffer from infections. And the recent pandemic has shown that very impressively, I think, as well. So aging is really one of the major risk factors for basically all of the disorders that are listed here, and especially also death. Now, if we would search for a unifying concept underlying all of the conditions listed here, we would find something like it's probably related to age-related systemic changes, which ultimately lead to something that people in the field call inflammaging. So inflammaging really points towards a systemic low-grade inflammation in elderly people. Now, during the time as a postdoc in Tony Weiss-Correy's lab at Stanford University, we were very much interested in analyzing these systemic changes and putting them in the context of brain aging. So basically what we did is, as a first start, we analyzed serum or blood factors to come up with a sort of an aging signature. And we did this for human blood as well as for mouse blood. And here you basically see a summary of these measurements where you can appreciate that in this non-supervised hierarchical clustering, the measurements of those 160 factors in the blood of humans perfectly discriminates the different age groups across the board in humans as well here in mice. The other interesting thing that we observed was that we have a progressive increase for about half of all the factors that we measured, again, in human as well as in mice. If we have a look at which of those factors are significantly correlated with age, we end up with a number of 38 factors for human and 18 of mouse origin. The really interesting factors came up when we looked which of those factors were actually shared between the two organisms. So in other words, would be species conserved. I would just like to mention two factors here, which are CCL11, also known as eotaxin, and CCL2, which is MCP1. And these two factors are very well known to be chemoattractants or cytokines for eosinophils and macrophages. So now you start to understand why we got pretty excited about eosinophil research as well. So here you see the plasma levels, the absolute plasma levels over different age groups shown for human cohorts and mouse cohorts. As you can appreciate, the levels come significantly up later in life, so pretty late, which pointed us towards the question, is this really a consequence or a cause of the aging process? And to address that question, we basically performed an experiment in which we tried to assess whether eotaxin CCL11 is a pro-aging factor which could accelerate an aging phenotype in young mice. So we systemically administered CCL11 into young 3-month-old mice and then assessed neurogenesis in the brain. So we looked at the dentate gyrus of the hippocampus. And we also performed learning and memory tasks with these mice. And just to summarize very briefly, this nice work conducted by Sol Vileda, who is now also a professor at UCSF, what we observed in this study is that the mice treated with CCL11 had a significantly decreased amount of new neurons that formed in the dentate gyrus. So basically, the CCL11 treatment phenocopied an aged mouse. Also, when we look at the learning and memory tests that we performed, we saw consistently that CCL11 treatment led to worse performance in the mice, either based on this radial arm water maze test, where the treated mice performed more or did more errors, and also in this contextual fear conditioning paradigm, where the CCL11 treated mice froze significantly less than the sham-treated mice. So these results pretty much confirm that CCL11 is a pro-aging factor. Obviously, there is much more recent work that has been published. There is all sorts of other different factors that are now considered to be pro-aging factors, for example, B2M. And also the plasma proteome has been analyzed in way more detail now, with over thousands of different factors in protein that have been assessed over different age groups. And this basically now allows also to predict the biological age based on different parameters that you can measure in the blood of humans. So these aging clocks have been established. For us, the finding of CCL11 as a pro-aging factor was super interesting, and obviously we wanted to know whether there is a specific source that produces or is responsible for this increased levels that we observe in aging. And so what we did is we simply screened different tissues. This is a Western blot that I show here for CCL11 and CCL2 expression. And what became immediately apparent is that white adipose tissue, so either subcutaneous or epididymal, showed the highest expression of these two factors. And if we zoom in into a cohort of young versus aged mice, we can also appreciate it that during aging or upon aging, these factors get significantly upregulated. So this basically told us that this pro-aging factor CCL11 has or is likely to be coming from the white adipose tissue. Now, from work from others, we already knew that adipose tissue is a very interesting, let's call it organ. It's no longer to consider solely to be an energy storage system, but it's a real endocrine immune organ. And very beautiful work by Richard Loxley and A.J. Chawla from UCSF have previously shown that in mice, if you have a lean homeostatic situation, that eosinophils and macrophages in adipose tissue sort of are in balance. And they have also shown that IL-4 derived from eosinophils is quite important to keep macrophages in an alternatively activated non-inflammatory status, which will then promote the tissue homeostasis and even sustain metabolic healthy adipose tissue beiging. Now, the picture changes completely if you go into an obesity model where sort of all of a sudden macrophages expand and take over. The IL-4 from the eosinophils is no longer sufficient to keep those cells in an anti-inflammatory state. And these macrophages, they become inflammatory. Now, in our own study, we wanted to see whether there are similarities or commonalities observed between obesity and aging in the context of this ratio of eosinophil to macrophage content in the white adipose tissue. So we basically dissected adipose tissue from young and aged mice and flow cytometrically assessed the frequencies of those cells, as you can see here. So if we quantify that, you can immediately appreciate that the frequency of eosinophils quite substantially drops in aged animals, whereas we see an expansion or accumulation of macrophages. And obviously the ratio between eosinophils and macrophages drops significantly with age. So this has been very closely associated with the occurrence of local adipose tissue inflammation. So if we look at pro-inflammatory markers in the adipose tissue of young versus aged mice, factors such as IL-1 beta and IL-6 are significantly upregulated. Now, once we saw these results, which are quite consistent with what people have seen in obesity models before, we were wondering whether we could somehow revert this imbalance between eosinophils and macrophages. And obviously, you have different approaches how you could do this. We took the following approach. We first performed a parabiosis experiment. This is a condition where you basically join the circulatory system of a young to an aged mouse. And obviously, you also do the isochronic pairings, young and aged as control. And then we basically, after 4 weeks of pairing, we then reanalyze the adipose tissue of these mice. And here is the flow cytometry data for each pair shown here. So this would be the young isochronics, the aged isochronics, the aged heterochronics, and the young heterochronics. And here is the quantification of these results. Basically, what's important to see here is that we get a partial rescue of the eosinophil frequency in the young or in the aged heterochronic mice, the macrophage frequency comes down, and obviously the ratio is partially restored. Now, if we look at the inflammatory parameters locally in the fat of these parabiosis mice, we can see that the inflammatory markers go significantly down if you compared old ISO to old HET across the board. Now, obviously, you could argue that upon parabiosis, a lot of different factors, not only cells, are exchanged between those mice. So we were looking for a cleaner system to phenocopy these findings. And basically what we decided to do was a transfer experiment where we injected eosinophils from young mice into aged wild types. And this is a first proof of concept study or experiment where we just injected twice eosinophils into aged mice. We did this intraperitoneally and then basically looked where do these cells end up. And that was a very surprising finding because what these results told us was that the eosinophils pretty selectively migrated into the adipose tissue, as you can see here. Those eosinophils were GFP labeled, and we could flow cytometrically look for the GFP signal across those different tissues. So that was very promising. So then we came up with the sort of real experiment where we decided to transfer continuously over 1 month, 8 times, so biweekly eosinophils intraperitoneally into aged wild-type mice. As comparison, we had young mice, HPBS-treated mice, and then we transferred 2 different sets of eosinophils. Those wild-type eosinophils in red, they come from an IL-5 transgenic background, though. This is what I have to mention. And then we also transferred IL-4 deficient eosinophils. So important to realize is that no matter whether the eosinophils are IL-4 deficient or sufficient, we see migration or restoration of eosinophil frequency, macrophage frequency goes down, and we have a complete restoration of the eosinophil to macrophage ratio in the fat of these treated mice. If we look at the local tissue inflammation, we can see that all markers that we assessed here, so CCL2, IL-1 beta, and IL-6 upon eosinophil treatment were decreased basically down to a level of a young animal. For IL-beta, this was not IL-4 dependent, but for the other 2 factors, definitely we had an IL-4 associated effect. Now, also on a histological level, we saw rejuvenating effects, if I may call those like that, in that the eosinophil treated mice showed reversed adipocyte hypertrophy. So they had smaller adipocytes again as compared to the HPBS control. Now, if we look systemically, if we analyze the blood, if we measure the same factors as locally in the adipose tissue, we saw an exact mirroring of what was going on in the adipose tissue. So again, CCL2, IL-1 beta, and IL-6 were significantly down in the eosinophil treated mice. And this was IL-4 dependent, again, for CCL2 and IL-6, but not so much for IL-1 beta. Now, obviously, we wanted to know whether eosinophil treatment could now affect age-related conditions. As we have shown, we can get rid of this inflammaging or this systemic low-grade inflammation. And we basically focused on 2 different conditions, which are frailty and immunosenescence or immune decline in aging. So we first measured physical fitness in mice. How did we do that? I want to show a short movie here. So obviously, I'm joking. We did a grip strength analysis and rotarod, so muscle force and endurance testing. And you see, this is the exact same treatment regimen that we had before. And what the results told us is basically that each and every mouse that has been treated with IL-4 sufficient eosinophils performed better after the transfer or the treatment than before the treatment. Whereas aged mice, sort of the control condition, performed significantly worse. And this was IL-4 dependent, this effect, because we saw no amelioration in the transfer of IL-4 deficient eosinophils. So here is the delta of pre versus post treatment for grip strength. And then the same picture was also true for the rotor rod, in which only the eosinophil treated mice performed better. We also showed that the eosinophils have to migrate to adipose tissue, because if we block their migration with pertussis toxin, we don't see this beneficial effect. So this is a protocol that has been very nicely established by David Furinger and has been published. So we also show that if we pre-treat those eosinophils to be transferred with pertussis toxin, we see less eosinophils in the fat, IL-6 levels are higher, and then grip strength is not ameliorated. Rotor rod is also not ameliorated upon transfer. As a last thing, I would like to show you some data on immunosenescence or immune fitness. Here we additionally introduced an ovalbumin immunization, and we basically assessed the hematopoietic potential of the bone marrow cells, as well as we quantified the B cell and antibody response upon immunization. For time's sake, because I'm running out of time, I'm going to skip the hematopoietic potential part here, but I'm quickly going to show the immune fitness in terms of germinal center B cells that we quantified here flow cytometrically after immunization. You can see that the germinal center B cells here, positive for PNA staining, negative for CD38, decrease with age. And you can basically restore that with eosinophil transfers. And this is a partially IL-4 dependent effect. If we do ELISpot assays, we can also see that we partially rescue the number of OVA-specific IgG producing B cells with the eosinophil treatment. And if we measure the OVA-specific IgG titers in the serum of those mice, we see the same picture. So the mice receiving eosinophils, they have higher IgG titers against the immunization. Are these findings relevant for humans? Yes, we think so. We have also shown in this study that eosinophils are actually present in human omental white adipose tissue. We did this by immunohistochemistry. We quantified over different age groups, and also here we see a negative correlation with age. We also did immunofluorescence, where we can nicely show that these eosinophils are in the fat of humans. We also established a flow cytometry protocol to quantify those eosinophils in the human blood. And also here we could confirm the negative correlation. And obviously, as a last thing, we also measured pro-inflammatory mediators in the fat of humans. And here we also saw a positive correlation for IL-6, IL-1 beta, and TNF, not so much for TNF, but IL-6 and IL-1 beta. So pretty similar as what we observed in mice, which could indicate that it's really a conserved biology here. But I'll stop here, and I think, or I'm pretty sure that Professor De Filippis will further educate us on eosinophils in human adipose tissue. So as a summary, important points are there is a loss of adipose tissue homeostasis, which leads to chronic inflammation. Adipose tissue is a major source of IL-6. Targeted treatments to reestablish homeostasis has rejuvenating effects. And we saw that eosinophil transfer has only beneficial effects if those eosinophils actually home to the adipose tissue. And these effects were largely or partially IL-4 dependent. So adipose tissue homeostasis might represent a really attractive therapeutic target. With this, I would like to end and thank all the people that have been involved in this study, especially our main collaborators, Dr. Marionotti and Dr. Carsten Rieter. Thank you very much for your attention. I'd be happy to take questions.
Thank you. That was a superb talk. And there are already several questions that have popped into the the Q&A. So again, if you do have questions, please go ahead and type those right into the Q&A. We have a couple of technical questions for you, Dr. Eggel. There's a question about how the CCL11 or the eotaxin 1 was administered in those mice.
So the CCL11, that was intravenous injection.
And there's a question about, in those CCL11 transferred mice, did you ever try to administer the CCL11 in an eosinophil deficient mouse to see if this is entirely due to the increased eosinophils or to CCL11 itself?
No, that has not been done. And as I mentioned, this has really been spearheaded by my colleague, Dr. Saul Vileda, who has now his own lab. I'm sure he followed up on these questions, but to my knowledge, eosinophil biology was not really in the focus in those studies. So the mechanisms underlying these CCL11 pro-aging effects is not yet solved.
Dr. Ackerman has a question here. Do any of the eosinophil deficient mouse strains show premature aging?
This is a really good question. We have never used DBL Gata mice, for example. I think they're very prone to develop obesity. I wouldn't be aware that anybody has aged them, past 12 months or more. But I'm not really aware that this has been done.
Okay. We also have a question from Jenny Martinson about how long does the vitalizing effect of your transferred eosinophils last?
Yeah, also that is a very good and interesting question. Obviously, this is very transient. So if we treat the mice for 1 month, we see the beneficial effects vanishing after about 2 months. So that, tells us that we need a sustained treatment if we want to go down that route. We think that this is partially attributed to the relatively short lifespan of eosinophils and that you have to replenish that pool over and over again. So maybe, coming up with an approach to deliver eosinophil-derived cytokines like IL-4 or IL-13, especially or specifically to fat, would be a more suitable strategy here.
And there was a follow-up there as well. Is there any correlation between your adipose eosinophil numbers and BMI that is maybe irrespective of age?
So that's also very interesting. And I drew some parallels between obesity and aging, but really, if we look at our aged mice, they're nowhere close to obese mice. So they're like 10 grams less heavy. So the fat content is definitely lower. And as I said, Richard Loxley's lab and H.H. Holla's lab, they have nicely shown that decreased eosinophil frequency correlates very well with the weight of those obese mice.
We also have a question from one of our other speakers. Dr. De Filippis is asking, did you look at the subcutaneous fat in these mice specifically compared to epididymal?
Yes, we did that. It's somewhere in a supplementary figure of that paper. And to make a long story short, we saw the most pronounced effects or rejuvenating effects in the epididymal wet as compared to, not so much going on in subcutaneous fat.
Dr. Voringer has asked, do you see any influence on the ILC2 levels after your eosinophil transfers?
Excellent question, Dr. Voringer. We're currently studying this, ongoing studies.
There are also several questions, and so let's see if I can sort of combine them together, but they're looking at the use of dupilumab. So are you familiar with dupilumab?
Absolutely.
So there are some questions about the use of dupilumab in young patients, particularly with severe atopic dermatitis. Do you think there are any biomarkers or chemokines we should be following over time, or any concerns you might have about the impact that this treatment would have on adipose tissue homeostasis? Yes,
also that is a very interesting and obvious question to ask after these results. Obviously, I'm not a physician. I would not make any, forecasts or recommendations here. I think it's really important to monitor closely what happens long-term upon dupilumab application. and maybe especially focus on what's going on in the adipose tissue. Yeah, that would be interesting.
Excellent. Well, we'd like to thank you again for a really fantastic talk.
Thank you very much for having me.
And if we could put the slides back up to move on to the next speaker. Thank you. So we are moving on now to jumping to cardiovascular disease. So we did warn you in the beginning, this is a very broad topic that we're covering. So we'll move on to cardiovascular disease, and then we will pop back into bioenergetics for the final speaker. So without further ado, we have Dr. Konstantin Stark. He's a cardiologist at the University Hospital in Munich. This is Ludwig Maximilian University. And he is going to be speaking about eosinophils as new players in cardiovascular diseases.
Okay, yeah, thank you very much for this kind introduction. We will stay in the field of inflammation and obesity and aging, but not the cause of aging and obesity, but rather the consequence, which are often cardiovascular diseases. So are there any genetic indications that myocardial infarction as one of the most common cardiovascular diseases and eosinophils are connected? So there is a study which showed that sequence variants affecting eosinophil numbers associate not only with asthma, but also with myocardial infarction. So this is kind of the first indication that eosinophils might be important in cardiovascular diseases. There has been also another study by colleagues from Germany, from Erlangen, and they showed that eosinophil-derived mediator, eosinophil cationic protein, ECP, is elevated in patients with cardiovascular diseases, especially stroke and ischemic attacks. And also, when they looked at the stage of atherosclerosis, they saw that especially in patients with advanced atherosclerosis, there are elevated markers of eosinophil activation. So how do eosinophils contribute to these diseases? Obviously, we've heard that they are involved in adipose tissue homeostasis. This could be one factor, which is not really well investigated, but it is known that eosinophils have a very strong pro-coagulant and pro-thrombotic effect. So it's known for a long time that eosinophils are a main source of tissue factor in the blood, and tissue factor is the main activator of the coagulation cascade, triggering then the formation of fibrin formation, which then leads to obstructive clot formation. And this paper showed that eosinophils have a very specific or particular lipid composition of the cell membrane, which fosters a very active form of tissue factor, which is normally inactive in the blood, because otherwise we would always have thrombi in our blood, which is obviously not desirable. But this specific form of the cell membrane in eosinophils promotes a very active form of tissue factor, which then triggers the coagulation cascade. And what is also evident is that diseases which have very high numbers of eosinophils in the blood are also associated with thrombotic complications, so indicating that eosinophils really have a strong prothrombotic effect. So do we see eosinophils in thrombi in humans? Yes, we do. So they are enriched in thrombi from myocardial infarction, as you see on the top, but they are also enriched in stroke thrombi. And if you compare these two entities, acute myocardial infarction and acute ischemic stroke, we see that they are even more enriched in stroke than in acute myocardial infarction. So indicating that they might indeed be involved in this process and not passively deposited there. So this is why we investigated if eosinophils contribute to atherosclerosis and thrombosis, which we published in a blood paper in 2019. So for that, we generated eosinophil deficient mice and crossed them with ApoE mice, so that we have eosinophil deficient mice which develop atherosclerosis, and compared the degree of atherosclerosis in these mice. So to our surprise, we saw that the mice which didn't have eosinophils, which were eosinophil deficient, the DBL gata mice, that they also had less atherosclerosis, as you see here on the right side. Then obviously the question came up, so how do eosinophils promote atherosclerosis? And for that reason, we looked into the plaques directly, and there we couldn't identify any eosinophils, which is consistent also to human studies where eosinophils could not be identified within atherosclerotic plaques. However, when we analyzed these atherosclerotic plaques, we saw that there are more macrophages within the plaque, there are more neutrophils in the plaque, so indicating that there's increased inflammation going on when eosinophils are present, and that there are more platelets adhering to these plaques. And we know that platelets are a main trigger driving the progression of atherosclerosis by recruiting other immune cell types. So the question is, how are eosinophils activated in atherosclerosis? And interestingly, we have here something which is an interesting connection to the previous speaker. We found that CCL11 is elevated in ApoE mice in response to high-fat diet. And this is a factor which is activating and has a chemotactic effect on eosinophils. There are also more eosinophil platelet complexes in ApoE mice, which are receiving a high-fat diet compared to the ones which do not receive a high-fat diet, so indicating that there are intense interaction between eosinophils and platelets. And as I've shown you, there are more adherent platelets in mice which have eosinophils compared to ones which don't have eosinophils. And one reason for that is probably that eosinophils activate endothelial cells, and there we could see that endothelial cells expose less von Willebrand factor, which is the main factor for platelet adhesion on the endothelium. And we could also show in vitro that eosinophils promote the exposure of von Willebrand factor from endothelial cells. Okay, so I've shown you that platelets are able to support plaque formation, but the next question is, do they also promote thrombosis, which then leads to myocardial infarction and stroke? For that, we performed intraocular microscopy. Here we used an injury model where we used ferric chloride and used labeled platelets and eosinophils with the EU cream mice, which we received from James Lee, which was crossed to a calcium indicator mouse. And from these images, we could see that there's a lot of interaction going on between eosinophils and platelet aggregates. And if we have a look at these calcium signals and eosinophils, we see that during interaction, which is marked by the blue boxes, there's a sharp increase in the calcium signal in eosinophils, indicating that there might be an activation going on during the interaction between platelets and eosinophils. When we look at the effect of eosinophils on platelets, we very often saw that eosinophils seem to form a nidus for the formation of platelet thrombi. As you see here, there's a eosinophil in the middle of a thrombus, which is growing over time, as we see here by intravitreal microscopy again. So we went on to investigate our our ApoE mice, which have atherosclerosis, and compared the ones which had eosinophils to the ones which did not. And there we saw that the ones which did not have eosinophils formed less thrombi within the carotid artery. So the question is, why do these mice form less thrombi? One factor here might be eosinophil extracellular traps. It has been described for neutrophils that they form extracellular traps, so expelling the DNA, which is decorated with granule proteins. And this process has also been described for eosinophils, but in other settings than thrombosis, mainly in inflammatory diseases. So that's why we checked for the presence of these eosinophil extracellular traps in thrombi, and we could see them in human as well as in murine thrombi. So you see here eosinophils marked by major basic protein, and here you see strands of DNA which are decorated by this granule protein, major basic protein. So next we asked how are these eosinophil extracellular traps formed, and we saw that direct contacts with stimulated platelets, but not with supernatant of platelets, induced the formation of eosinophil extracellular traps, so indicating that there's kind of a mutual activation of eosinophils and platelets in thrombosis. What are the factors triggering the formation of EETs in our setting? We can block them by blocking the interaction between platelets and eosinophils by an anti-P-selectin antibody, or interestingly by using an anti-cyclic 8 antibody. When we used this anti-cyclic F antibody in mice, which is known to deplete eosinophils, but in this case we applied it very shortly before we induced thrombosis, so only 30 minutes before, and this did not lead to a significant reduction of eosinophils. We saw that this reduced also the formation of thrombi, but also resulted in less formation of eosinophil extracellular traps. So how do these eosinophil extracellular traps interact with platelets? We found in vitro that these eosinophil extracellular traps are decorated by major basic protein, as I told you before, but in vitro we saw that exactly at the spots where we saw enrichment of major basic protein, we also saw a lot of platelets on these eosinophil extracellular traps, so indicating that they might form a scaffold and an activating factor for platelets. And that's why we checked the blood in MBP deficient mice and saw that whole blood aggregation is decreased in mice lacking a major basic protein. And to come back to the in vivo situation, we also performed our arterial thrombosis model in carotid arteries of mice, and saw that also there the formation of arterial thrombi is decreased in mice which don't have major basic protein. So to sum up this part, we think that there's a mutual activation of platelets and eosinophils. So platelets foster or induce the formation of eosinophil extracellular traps, which are decorated by MBP, and this in turn fosters the activation of platelets. So in what is our model of the role of eosinophils in atherosclerosis? I told you that we don't see eosinophils within the plaques itself, but we see that there is an upregulation of CCL11 in atherosclerotic plaques, which probably activates eosinophils, which form aggregates with platelets, and this results in mutual activation. Activated eosinophils leads to the release of von Willebrand factor from endothelial cells, which in turn foster platelet recruitment, and these platelets then support the recruitment of monocytes and neutrophils, which increase plaque formation. In arterial thrombosis, which is a result of atherosclerosis, we see that there is plaque rupture, which then leads to the adhesion and aggregation of platelets. And these platelets have several effects on immune cells. They have not only effects on eosinophils, but also neutrophils, which also have a thrombostabilizing effect. But focusing on eosinophils, we see that platelets activate eosinophils, which then form eosinophil extracellular traps. And these are decorated by major basic protein, which in turn fosters platelet aggregation. But as I told you in the beginning of my talk, eosinophils also have a very active form of tissue factor, which then leads to thrombin formation and the formation of fibrin, resulting in an obstructive clot and myocardial infarction or stroke. So I would like to thank you very much for your attention, and obviously the collaborators which contribute to this, especially James Lee, my team here in Munich, especially Charlotte Marx and Julia Nowotny, who worked on this topic, but also all the others. Thank you very much.
Thank you, Dr. Stark. That was an excellent talk. I loved the videos, especially of that model. We do have a technical question. Do you know who is producing that elevated CCL11 that you're seeing in the ApoE mice?
Yeah, that's a good question. So it seems like it's smooth muscle cells, but we haven't specifically looked into that.
Okay. And we have a question from Dr. Rafus. The intact eosinophils were not found among the inflammatory infiltrates when you were looking at the plaques, but did you look in those plaques for the presence of eosinophil-derived granule proteins?
No, we haven't. That's a good point. I mean, it could be that we don't detect our eosinophils because they are so short-lived in the tissue, and there are granule proteins there, but we haven't looked for that.
Okay. And so when you take away the eosinophil extracellular traps, you appear to lose the stability of those thrombi. And certainly some of that is through the MBP in the traps activating platelets. But the question appears to be, are you saying that the neutrophil-derived traps that would still be present in the absence of eosinophils are not sufficient to kind of maintain that stability? And did you have any thoughts or comments on that?
So we think that the eosinophil extracellular traps are per se without the MBP not sufficient, yes, because probably the MBP is the factor which fosters platelet aggregation and activation, and this is probably the factor which then stabilizes the thrombus.
There's a question from Dr. Cleon. You showed that eosinophil deficient mice have less or lower ASCVD, but what about IL-5 transgenic mice? Would they have more?
Good question. I haven't looked into that. Yeah, that's something we should check. That's true. We haven't looked into that.
Okay. Dr. Ackerman, I love his expertise across such a range of topics. He is mentioning that there is a literature on MBP affecting coagulation pathways. So he's wondering if you have any comments about that. And he mentions specifically the kallikrein system.
Yeah, there are some, let's say, papers that it's also activating the clotting system. We haven't looked into that because for our setting, we think that platelets are more important. But true, it could be also something which contributes to the lack of thrombus formation in our mice.
There's also a question referring to Dr. Shee's work from Brigham and Women's Hospital, who has published on a cardioprotective role of eosinophils following myocardial infarct. So do you have a sense of, or can you comment on where your story fits into a broader literature, or perhaps of roles of eosinophils in fibrosis?
Yeah, so honestly, I was quite surprised by this rather beneficial effect of eosinophils, because yeah, I mean, it's probably a question of the model. So I mean, they used a chronic left anterior descending artery ligation model, which is probably another thing than we usually see in daily practice, where we try to establish blood flow again in patients. Yeah, I was honestly quite surprised, but I expected the opposite, to be honest, in this model. Also in context of the fibrotic effect of eosinophils and other diseases.
There is a question, and I apologize if I'm mispronouncing this name, from a Dr. Ramakrishnan. He would like to know if we should be using eosinophil depleting antibodies like benralizumab to treat acute coronary syndrome.
Yeah, honestly, we also thought about that. I think it's too early to really do that, but we were also discussing if this might be something, if we think that it is beneficial in atherosclerosis and thrombosis. I mean, the drawback, as you mentioned, is that there are 2 studies which showed that depleting eosinophils is rather detrimental in acute myocardial infarction on the long term regarding tissue fibrosis. So I think that the question is, if we induce a ischemia reperfusion injury, not a chronic LAD ligation, is there a beneficial effect of eosinophils? Then I would be more confident to also recommend this.
All right, well, thank you very much. That sparked a lot of interesting conversation. And if we can go ahead and pull the slide up for the next speaker. This is our final speaker of the day. This is Dr. Elena De Filippis, and she'll be speaking about adipose tissue resident eosinophils and insulin resistance. Is there a human connection? And although she is originally from Italy, she is currently an assistant professor of medicine and also the associate research chair of the Division of Endocrinology at the Mayo Clinic, Arizona.
Thank you, Lisa. I'd like to thank the organizer for putting together such an exciting session. I've learned a lot, and I'm really happy and excited to share also our experience. So with any further delay, I hope you guys are able to see my slides. And so as Dr. Spencer mentioned, I will shift the focus and go back on adipose tissue. And for this presentation, I do not have any financial disclosure. And what I will focus today is specifically bringing back your attention on obesity, the role of adipose tissue in health, so in insulin sensitivity state, but also sharing with you the consequences of what we observe in human subjects that deal with obesity in terms of adipose tissue content of eosinophils. And so Dr. Eggel, as I mentioned, as well as Dr. Stark, that obesity is indeed a complex disease and affecting multiple organs. Sad to say that unfortunately, at least in the United States, today, as many as even 1 in 3 adults are battling this disease. And this is definitely not an inexpensive illness. It's been estimated that about 21% of the American healthcare expenditure is devoted to either treat obesity or its complication. In terms of complication, really on the left panel of the slide, you can see listed many diseases, including insulin resistance, type 2 diabetes, excessive accumulation of fat around the liver, which leads to non-alcoholic fatty liver disease that, if progressing and arriving to cirrhosis, is now becoming the #1 indication in the United States for liver transplant. And there are cardiovascular disease, as the previous speaker outlined, is often associated with obesity, and there are even many cancers that are now recognized to be associated with such disease. Now, obesity clinically, and today we'll talk mostly about patients, so I want to make sure that we are all on the same page. And if it's true that we define obesity as an excess of weight and accumulation of adipose tissue, we also clinically use what's called the body mass index, which is the relationship between your height and your weight. And when these two data are put together, the number, if it's less than 25, that identify as subjects that we consider to be normal weight, as long as the BMI is greater than 19. And so we call those lean subjects. But if this computation leads to a number greater than 30, then we define medical obesity. And when it's higher, we then break it down in different classes. However, for the purpose of today, we'll focus, and you'll see most of my data presented as either referring to lean less than 25 or to obese more than 30. So Dr. Eggel has already mentioned and alluded to the fact that originally adipose tissue for many years was considered simply to be a dump of excessive energy under the form of a lipid droplet, so triglycerides stored and nothing more. However, in the 90s, the discovery of some molecules, now today referred as adipokines, led to a shift in the concept of adipose tissue. And this is a tissue that now we consider fully to be an endocrine organ. And in fact, by releasing some of the molecules, some of which are listed on the left side of this slide, we have now learned through many years of research, both in animal as well as in humans, that these molecules do affect the metabolism of peripheral tissue. One example in this slide is adiponectin, that when is released, act on both, on all these following tissue, on the liver, on the muscle, as well as on the pancreas, and impacts the metabolism of each of the single organ in a nice and organized system that leads to what we define often as insulin sensitivity. Now, it is and leptin is another important peptide and hormone that now we are aware of in healthy adipose tissue, being a food regulator as well as an energy expenditure regulator, and having also many more effect and actions. However, previously has already been mentioned that fat tissue is not just simple accumulation of adipocytes, but there is the presence of other cell system, and particularly the immune cell system have been depicted interspersed among adipocytes. And the relationship that there is now between immune cell as well as adipocytes is growing in understanding, however, is not fully completed. So what happens when a person is gaining weight, the first thing that occurs is the adipocytes do expand. But that's not just a mass expansion, it's also a change in function. And so in people that are gaining weight, we know that leptin level goes significantly very high, and adiponectin level drop to very lower, as well as there are other pro-inflammatory markers that are increasingly produced and released. But as you heard, there is also a shift in the immune cell population with a more representative presence of macrophage with the pro-inflammatory features. Other also cells have been described, such as a change in lymphocytes, and even endothelial cells do change the expression of adhesion molecules. All of these leads to what is now being known to be a low-grade chronic inflammatory state, which is also very indicative and distinctive of obesity. And this is not just limited to the adipose tissue. We know that all these mediators are released in the bloodstream and do affect the action and the metabolism in different organs, such as in the liver, in the muscle, as well as in the vessel, as you just heard from Dr. Stark. And so all of this together really leads to what we call the insulin resistance syndrome, which is defined often as a reduced sensitivity to the action of the hormone of insulin, which translates in many metabolic impairment. So starting and re-mentioning what others have done before me, Dr. Loxley and Wu, early about 10 years ago, published the first observation that among the immune cell system, eosinophils were probably protective of insulin sensitivity, and a detrimental effect of weight gain, at least in an animal model, was shown by noticing a significant decrease in eosinophils count as resident cell of the adipose tissue. And then the use of two genetic models show that whether there is an excess of eosinophil presence, the glucose load are actually improved in this animal model when they are compared to wild type, and the opposite happens if you use a knockout genetic animal model. We took these and created our own experience, which I will only briefly touch today, and generated a specific model where we overexpress human eotaxin 2 only in white adipose tissue. Therefore, we actually accumulated the eosinophils only in the white adipose tissue. And our mice on 12 weeks of high-fat diet demonstrated a significant less accumulation of adiposity as represented as total body weight and percentage of fat. And when we challenged those mice with a glucose load, their blood glucose in circulation were significantly lower when compared to our wild-type animal model. In addition to that, going back to the adipose tissue health, we showed the persistence of an elevated adiponectin with a lower instead leptin level, typical of a more healthier animal. And then accumulation of triglycerides, so excess of lipids in the liver, was decreased in our animal model. So was also the inflammatory profile, which was more of an anti-inflammatory marker identified in the adipose tissue that we examine, and these are example and data from subcutaneous, but we had similar results from perigonadal. So starting with this hypothesis, the next step clearly is we treat patient in clinic, what is happening to our patient with obesity. And so we hypothesized that similarly to our experience in mice, when we would be looking at adipose tissue coming from subjects with obesity and insulin resistance, we would be finding a decreased amount of eosinophils, and we felt that that reduced content of eosinophil could be affecting adipose tissue biology. So how did we try to answer our question? We collected subjects spanning in a different age range, but not really older or very young, and either, as I said, lean subjects or subjects with obesity without too many other comorbidities. And we collected blood, performed oral glucose tolerance tests together with the subcutaneous fat biopsy. So far, our cohort is equally distributed between lean people and obese people. And I want to draw in this table your attention to the fact that by design, of course, the BMI of our subjects is higher in the group with obesity. They also have evidence of insulin resistance represented by a mild but still significant increase in systolic blood pressure, fasting blood glucose, determination of what we called OMA-IR, one of the indices that we measure for insulin resistance, was significantly elevated in the subjects with obesity. And as described over and over in literature, our subjects clearly had an inflammatory systemic status, as shown by the elevation of highly sensitive C-reactive protein. They had also high level or higher level of triglycerides with the lower level of HDL, again, known features of insulin resistance. Another typical association with insulin resistance is the elevation in circulation of neutrophils. So this data didn't surprise us. When we look at eosinophils in circulation, we found absolutely no significant difference between these two groups. So further evaluation of the plasma detection of this adipokine, again, and similar to what happens in animal model with wild type, the adiponectin was significantly decreased in the subjects with obesity and was also showing an increased level in leptin in the plasma. Well, these two adipokines come specifically from the adipose tissue. So when we collected their subcutaneous adipose tissue and looked for the mRNA, it was no surprise to us that this picture was actually mirrored in the mRNA level, with a decrease in adiponectin and an increase in leptin mRNA level. And when we checked some of those markers for inflammation in the adipose tissue, we found several of those to be significantly increased. So typical subjects group that we were dealing with was definitely confirmed to be what also in the clinic sometimes we referred as prediabetes. Now, as others have shown before me, if you take a section of adipose tissue, you can definitely identify eosinophils. This is through the staining of eosinophil peroxidase, an antibody that was kindly gifted to us by Dr. Jacobson, our longtime collaborator. And so it's hard, though, to quantify eosinophils in this situation. So we took upon ourselves setting up a fax-based protocol that was until then not available in literature, and we decided to start by creating a scheme starting with the blood sample and translating that in subcutaneous adipose tissue, and using the same antibody scheme we actually were able to isolate this successfully eosinophil. Therefore, this allowed us to definitely quantify more precisely the amount of adipose tissue content of eosinophils in our subjects. When we did that and we expressed this as a percentage of eosinophils for C45 positive cells, we found that, like Dr. Wu, in subjects with obesity, the number of eosinophils locally is significantly decreased. Not only that, but alluding to an answer of one of the questions that was asked earlier, we actually now know that for every point increase in BMI, there is a significant decrease in eosinophils percentage content in the fat. And even more, the reduction in eosinophil percentage content in adipose tissue is tightly related to an increase in insulin resistance. So this is the beginning of our question: what is happening? What is the role of eosinophils in adipose tissue? Why there is this tight correlation? But even so, why eosinophils are decreased in adipose tissue? We try to look at some answer for this question, and we started by collecting eosinophils from the peripheral blood of our subjects, both lean and obese, purify them and isolating mRNA, and looking for cis chemokine receptor 3, which is the known ligand for one of the chemoattractant for eosinophil in tissue. So the idea here was, is the system that is known to favor a migration of the eosinophils into tissue somehow impaired? And to our surprise, we found the CCR3 mRNA level is decreased. Now, mRNA level often doesn't give us the full answer, so we took this to a step further. And what we did is, in peripheral blood of our subjects, we isolated using the MFI, and we were able, using this technique, whether we express this data for EMR1 or cyclic 8, to confirm that this also at a protein level is a consistent data, and subjects with obesity indeed have a decreased expression of CCR3. Well, the other part of this system is looking at the eotaxin, which is known to be expressed in both pre-adipocytes and adipocytes. So we looked in our subjects, and eotaxin 2 is what we use in our animal model. However, we know that there are 3 isoforms, and so we wanted to be complete and look all the 3 isoforms. And what we identified is, in our subjects with obesity, there is definitely a decrease in eotaxin 1, eotaxin 2, but we don't find quite yet a significant decrease in eotaxin 3. So what is the role of eosinophils in metabolism? So far, we have answered some of the questions. So we have a decreased level of eosinophils, and we have at least some indication of why the eosinophils may be decreasing in adipose tissue. So we went back to the animal studies and again to the experience of Loxley and Wu. And in the original paper, by using a reporter mice in perigonadal adipose tissue, which in the animal is the equivalent of our intra-abdominal adipose tissue in humans, they demonstrated that the majority of interleukin-4 that were able to be there was actually coming from the eosinophils. In a different animal study, we have in literature, there is knowledge that low levels of interleukin-4 are actually associated with a high level of leptin. And if you remember, our animal experiences show that our increased account for eosinophils in adipose tissue was leading to a decrease in leptin. And in this same study, when they administered the interleukin-4, this actually restored lower levels of leptin, allowed some reduction in adiposity, and improved the glucose metabolism. This was quite exciting for us. And then coupled with the knowledge that in population-based study, interleukin-4 polymorphism was associated with type 2 diabetes and low HDL. There is also a small experience in literature using cell culture that treatment with interleukin-4 can change and affect adipose tissue or adipocytes metabolism and also increase ATP synthesis, a form of energy. So we went back to our cohort and we asked the question, what is the interleukin-4 in the plasma of our subjects with obesity? And we found that there was a significant decrease in circulating level of interleukin-4. But as I told you, we collect subcutaneous fat biopsy of these subjects. And for those of you that don't work with this tissue on a daily basis, I want to remind you that the adipose tissue goes through a collagenase digestion, and that separates two phases, a floating phase, which is usually referred as adipocytes, and a phase that can be simply be collected at the bottom of your tube, and it's referred as stroma vascular fraction as an heterogeneous representation of many cells from immune cell to pre-adipocyte, mesenchymal cell, and others. And when we look at mRNA expression in these two fractions, we found that there was a significant decrease in interleukin-4 mRNA expression in broad fraction, and above all in the stroma fraction. So we collected the human primary mature adipocytes, and we were able to put them in a culture, and without going into too many details for sake of time, we demonstrated that even on a cell culture level, these adipocytes do retain features of insulin resistance with an increased leptin level. So what we did is we treated these cells with interleukin-4 and 13, and we were excited to find the mRNA expression of the leptin gene was significantly decreased in both primary mature adipocytes as well as a whole fat subcutaneous adipose tissue. Now, we also collected the blood from patients, and we stimulated with the cocktail that's known to increase and activate eosinophils. Specifically, we look at the amount of interleukin-4. We found a significant increase in their conditioned media. So we used this conditioned media on top of the primary mature adipocytes of our subjects with obesity and tested again the question, what happens to the mRNA level of leptin? And we found these again significantly decreased. So one of the last experiments we did more recently was, in addition to stimulate with an interleukin-4, we also use an interleukin-4 receptor alpha antibody. And what we observed is that in the adipocytes, looking at mRNA, there was not only a significant decrease in interleukin-4 mRNA level when only interleukin-4 was used, but when we look at the leptin level of the mRNA by treating the cells also with the antibody, such effect was actually blunt. And this was not only at the level of the mRNA, but it was also at the level of the protein released in the supernatant. So to summarize, We now have some evidence that, similar to our animal data, subjects with obesity and insulin resistance do have a decreased level of adipose tissue EOS. And it seems that in part the decreased content may be related to a problem with the CCR3 eotaxin signaling system. Whether this is the chicken or the egg, more studies are necessary. And we do have some evidence that the presence of eosinophils in adipose tissue does impact adipose tissue biology, as I show you in different fashion using different tools. Now, more studies obviously are needed, and our laboratory is focusing on additional evaluation of this interaction, as well as also different metabolic stimuli and how they impact the eosinophil function and migration, as well as how weight loss may restore dysfunction. I would conclude here by saying thank you for your attention, but also this work cannot be done without the tremendous help I have in the lab from a postdoc and my technician, but also I would like to spend a word about my longtime collaborator, friend, and mentor, Dr. Jacobson, together with my other group of friends, and also I can't forget the funding. And with that, I thank you. Thank
you. That was excellent. And just to remind everyone, please go ahead and type your questions into the Q&A. The chat is not going to bring those questions up for me, so go ahead and plug them into the Q&A, and that would be great. So just a few questions that you may or may not have the data on, but do you have a sense of how long eosinophils survive within adipose tissues?
That's a very good question. We don't have currently any data in humans. We know we did similar experiment of what Dr. Eggel was mentioning earlier. And for us, the survival in adipose tissue in animal is very, very short. So clearly, this could be the other side of the problem, but we have not been able to find the answer to such question, but it's a great question.
And you do have that great method, your flow-based method of being able to isolate those adipose tissue eosinophils. Have you, or can you share, have you been able to see any specific markers that might give you some insights into the type of heterogeneity we're looking at here? I know that's a
yeah, that's a great question, and actually brings a very warm feeling, because it's exactly what we are actually doing right now. We're able to isolate these eosinophils, and we know that they are viable. We already have done some epigenetic signature analysis. I don't have final data to share with you, but that's exactly one of the aspects that we're looking at.
And one other question, again, is maybe asking you to look into the future. But if you think about this fact that this low-grade inflammation that seems to be associated with obesity is obviously not a good thing, do you have a sense of how quickly, if at all, that could be reduced or
right? So in part, so to answer that question, we are enrolling subjects. And so one of the ideas is we have a group of subjects that's undergoing weight loss procedure, and weight loss is known to restore to some effect adipose health. So we are looking and tracking these in humans. However, based on the published literature, we know that in animals, 3 days of caloric restriction was able to restore eosinophil content in adipose tissue. These are data out of Vanderbilt experience. We have now looked in our animal model. We're very kind of focused on the human, and would like to know, so we're doing a time course there, and we would like to know whether we can answer this question.
Dr. Duda Denberg has asked, are patients with HES or hypereosinophilic syndromes generally lean?
So that's something else we're looking. It's hard to say what's happening, because we are trying to strategically stay away from recruiting those subjects and moving forward. However, those subjects, for example, I'm thinking of the EOE patients, they often have other problems that may interfere with their body weight. So in subjects, for example, that have problems with the GI tract, how can we discern easily whether they are lean because of their underlying disease or because they don't just gain weight? So it's an interesting question, but it's going to be hard to tease out. As far as the asthma, for example, there's a long experience that obesity does worsen asthma, but whether it's the eosinophilic asthma or not, I think there is a lot of studies that needs to be completed there as well.
Dr. Denberg came up with a follow-up question here. Would treatment of HES patients then be expected to cause changes in their metabolism?
So it's a very good question. It's actually an interest of us. We have an IRB approved protocol right now, as well as we are designing more study in collaboration with our colleagues in allergy and dermatology to try to answer this question, because based on the literature data on any of the drug that has been recently approved by FDA, unfortunately, metabolic markers are not presented, are not looked at. And so there is very little limited experience currently published that we can find on those large trials.
Dr. Rafos would like to know if there are any hypotheses on why CCL11 or eotaxin 1 is reduced in obese subjects.
So we are trying to answer this question again, this could be the chicken and the egg, right? It could be, so we're trying to answer this question by seeing what happens by exposing the adipocytes and pre-adipocytes to different metabolic stimuli. So if there's something that's happening with the transition on overweight and obesity, that's actually leading and impairing that signaling, or if it's something that's genetically present there and we don't discover until the problem has already been established. And so those are the things. So right now, the study are too early to give you a final decision on which one of the two may be the case.
That's exciting that that's to come. Dr. Nazaroff has asked, you see a decrease in IL-4 during obesity. Do you also see an increase in type 1 inflammatory mediators? And how do you think that might play into eosinophil migration into the adipose tissues?
So as I showed the data, we do see, and this is not just our experience, is a known reported experience in literature that there is definitely an increase in the Th1 markers of inflammation locally in adipose tissue. And that could be very well something that is affecting the migration of it. However, it was interesting to us to find that even eosinophils into the blood circulation do show some deficits there. So I think the question is the answer is going to be a little bit more convoluted than just a simple local effect of the adipose tissue inflammation.
So there's a comment here by Dr. Cleon. We looked at the change in BMI with benralizumab, and this was published in the supplement for the New England Journal of Medicine article, and they saw no effects. So just a comment there that Dr. Cleon added. Dr. Ju asks, what is the function of eosinophils in healthy adipose tissue, and why are they there in the first place?
So I do not have the final answer. Our current model and hypothesis is that we have noticed that the changes we see in leptin mRNA level as well as protein do not occur in lean subjects. So our working hypothesis right now is that the presence of eosinophils in healthy adipose tissue works almost like a brake and holder in check for the adipose tissue function. And as you remove that eosinophil brake, then situation or stimulus can lead to a higher level of transcription and secretion, for example, of leptin. And we know that high leptin, unfortunately, not only is a signature of obesity, but has also some metabolic impairment. And so the idea in the working model is what happens to the situation if we would be able to restore the eosinophils content. Can we actually bear down even slightly the amount of leptin and potentially other adipokines, which is the next step of our study in try to expand the potential interaction with eosinophils. So that's our current working hypothesis.
Excellent. Well, this has been a really incredible session. So I would like to thank all three speakers, and I wonder if Dr. Rufos is hopping back on to sign us off. There she is.
Here I am. Thank you very much, Lisa. This has really been a fascinating session. I think everyone will agree that we saw some very interesting observations in mice, and it's really exciting that we're beginning to see data in humans as well. So I would really like to extend very warm thanks to all three speakers. These were very clear and very, very eye-opening talks. I would also like to thank Lisa Spencer, who has put together this very original webinar. So thank you very much to everyone. Thanks also to attendees, because the discussion has been very interesting as well, very lively. I think that we have a next slide just to remind you that we're still offering free trainee membership. So anyone who has joined this series of webinars for the first time, if you have trainees that would be interested, this is really the time to engage them in our society's membership, because registration, membership is free. And with the next slide, I'm just going to remind you that we do hold these webinars on a monthly basis. We've been doing that since the month of January this year. The next webinar is going to be on the 15th of December. This is an exception to our general rule that we hold our webinars on the last Wednesday of each month, and that's because of the end of year holidays. That webinar will be on eosinophil gastrointestinal diseases. And we also have a webinar planned in January that will be held jointly with the European Mast Cell and Basophil Research Network. It should also be a very interesting collaboration with that network. Next slide. And so this is just a last slide to remind you about our society, to let you know how you can reach out to us, either through the website. You already have the link, I think, from the very first slides that we showed you. But this is also our email address if you wish to have more information about programs, or if you have any suggestions or a wish list about future topics that you'd like to see covered in the setting of these webinars. Thank you very much, and I hope to see you again at the during the November webinar. Bye-bye.